The technique in which we're using the living organism for the development of products, this is known as biotechnology. Biotechnology is a huge field in which we use different kind of microorganisms for the production of drugs and other materials. So biotechnology works for the welfare of mankind, works for the human welfare. So another point I want to explain that if the patient have diabetes mellitus, how can we treat? So firstly we develop with the tools of biotechnology. The tools, it's a process in which we synthesize the human genome for the repair of the cell. For example, diabetes mellitus. In diabetes mellitus, the patient blood pressure increases every time, and the destruction of the beta cell into the pancreas. So biotechnology helps us to diagnose the treatment of diabetes of the patient. Here, different tools in biotechnology. One is endonucleases. Restriction endonucleases. First, in this step, we cleave the DNA double-stranded molecule into a smaller erased fragment. The second one, which is DNA cloning, we replicate the DNA into one or more. Where we replicate the DNA? It's into the cell. Third one, which is probes. In this process, we cleave the DNA because DNA is a long strand, larger strand. We cleave the DNA into our nucleotides, into one to more nucleotides. Fourth one, which is southern blotting. This technique very important because due to these techniques, we arrange the nucleotides. We replace one nucleotide to another nucleotide. Rearrangement of nucleotide. Fifth step, which is restriction length polymerase. In this step, we identify the human genome. If we didn't identify the human genome, how can we repair the damaged cell? How can we design the genome? So this technique helps for the identifying of the genome. Sixth process, which is polymerase chain reaction. In this reaction, there is synthesis of millions nucleotides. With the help of virus and bacteria. Seventh, gene expression analysis. From these tools, we analyze the messenger RNA and protein products in all kinds of organisms. Another point, which is gene therapy. In this process, we transfer the genome into the disease that helps for the repair of the damaged structure. Gene therapy. And the last one, which is transgenic animal, this process very helpful because from this techniques, the transfer of genome from one organism to another organism. This technique is used for the production of organisms transgenic animals transfer from one to another, from organism to organism during replication. Biotechnology, very interesting field. If you have interested, if you want to analyze the structure of the bacteria in the test tube, you can take the microscope and study about the structure of the virus and different kind of bacteria, streptococci, and staphylococcus. This kind of bacteria, guys, examine with the help of microscope.